Welcome to Straight Science, everyone. Straight Science is an evening seminar series put on by University of Alaska Fairbanks Northwest Campus in Nome and University of Alaska Fairbanks Alaska Sea Grant. And tonight you're in the home office on a very warm, rainy spring night in Nome. And tonight's speaker, since we're warming up and we're getting ready for the season ahead of us, the summer season, this is a pretty timely topic and one that has affected this region in the not too distant past. So tonight's speaker is Sarah Schoen. She is a biologist with the U.S. Geological Survey, which, though the title may throw you, they have research responsibility for seabirds and other birds as well. Is that right? You'll let us know. And Sarah has been working throughout the state on harmful algal bloom events and die-off events, and she is going to give us a look at her results from her very extensive work on seabirds in Alaska on the topic of harmful algal bloom topics. And she does have forage fishes in there, and I'm grateful for that because we don't want to just be one-sided in the story, right? We want to hear the whole thing from A to B. So looking very forward. I'm hoping you have nothing but good news for all of us in the Bering Strait region, but we'll let you tell your tale. Take it away, Sarah. We're so glad you're here.
Awesome. Thanks, Gay. So I want to acknowledge all of my collaborators and co-authors on the talk from U.S. Geological Survey Alaska Science Center, Yumi Uramitsu, Danielle Garrick, Naomi Bargman, John Pyatt, Matt Smith, and Caroline Van Humert. And Danielle gets an underlined because she is the last woman standing with me here at USGS. And then also collaborators at the USGS National Wildlife Health Center. So a quick data disclaimer, some of these data are preliminary and subject to revision. And before I get started, I just want to give a quick background about myself. I grew up on a beach in Juneau without a road to my house. And to get to and from our car, we had to walk the beach or go on a skiff. We knew the resident herons by name, and one of my first words was whale. My dad was a biologist, and it was this upbringing that really fostered my interest in the marine environment and in conservation. And I was lucky enough to land a job as a biologist with the USGS Alaska Science Center in Anchorage in 2012. And I've been with the Seabird and Forage Fish Ecology program since then. I mostly study colonially breeding seabirds and forage fish and oceanography. And then starting in about 2016, I shifted towards a focus in seabird die-offs and harmful algal blooms. The USGS is a science research agency, and we provide science information to stakeholders, including the public, federal and state agencies, industry, and co-management councils. And we don't have any regulatory authority. So in this talk today, I want to take you on a journey with me as we work through some of the big questions that I've had about the impact of harmful algal bloom toxins in seabirds across Alaska. So being a seabird biologist in Alaska, the summer is my primary field season. But in the winter of 2016, I got pulled back into the field abruptly. As you all have probably been aware, there was a large marine heat wave that started in 2013 and 2014 and carried on for multiple winters. It was called the blob, colloquially. And that was the largest marine heat wave ever recorded. And in the winter of 2016, I got a call from my former boss, John Pyatt, saying that Dave Irons from the Fish and Wildlife Service had counted nearly 8,000 common murres on one beach alone in Whittier and Prince William Sound. And he told me I need to get out into the field as soon as I possibly could and try to count and figure out how many birds had died and how far that die-off stretched and determine the cause of death. So I quickly gathered a field crew and we set off to try to determine the size and the scope of the die-off, as well as the cause of death. So this die-off turned into be the largest seabird die-off ever recorded around the world. It stretched from Alaska to California, and we originally estimated that about 1 million MERS had died in the die-off, and that was updated recently by Heather Renner's paper to be about 4 million birds dying in the die-off. But we wanted to know why they died. And when we got into the field in cold, rainy Prince William Sound in early January of 2016, it was really apparent that these birds were skinny. You can see in this picture, their breastbones were sticking through their feathers, and the muscle had atrophied around their breast. So they had really lost a lot of muscle mass. And when we cut them open, we found that they were completely emaciated, no fat. You can see in this picture of the inside of the skin. Normally it would be yellow with fat, but it was just completely devoid of fat. And they didn't have any food in their stomachs either. So we knew that they'd starved, but we didn't know why they had starved, if it was just a lack of available forage fish prey or if it was something else. And soon after, Kathy LaFay published a really seminal manuscript about concentrations of different harmful algal bloom toxins, domoic acid and saxitoxin, found within harvested and beached marine mammals across Alaska. So in this figure from her paper, you can see the individuals that had saxitoxin detected are color-coded in purple. And following this and some other input by folks at NOAA and others, we plan to test die-off MERS from this event for HAB toxins. And briefly, to let you know a little bit more about HABs, if you're not already aware, harmful algal bloom toxins, or HABs, have increased alongside ocean warming, and they're characterized as events with very high or uncontrolled growth of toxin-producing algae. And these can harm a broad range of marine organisms, from fishes to birds to marine mammals and even humans. Saxitoxin is the main player in Alaska. I'm just going to focus on that today. And that's produced by the dinoflagellate Alexandrium. It can induce paralytic shellfish poisoning, and it has a regular historic occurrence in Alaska, although historically it was more in the Gulf of Alaska and Southeast Alaska than farther north. And at this point, there was very little known about HAB toxins in seabirds or their prey. So we wanted to put these die-off bird tissues into context. So my group and I, live captured and collected presumably healthy live birds from their breeding colonies in Lower Cook Inlet, which was near the epicenter of this MER die-off in 2016. And we also collected forage fish around Prince William Sound and Cook Inlet to try to get an idea of toxin levels in their prey. And we used a variety of methods for that. And then we also collected blue mussels, which are a really good indicator of environmental levels of saxitoxin in the environment, because they take up the toxin quickly and then they release it relatively quickly too. So our analytical methods included using commercially available ELISA kits, and these had been developed using shellfish toxins. So we had to validate that they were able to analyze seabird and forage fish tissues, and we did that in a collaboration with NOAA at NCOS. And then the analysis entails homogenizing 1 to 5 grams of seabird tissue or whole fish and invertebrates, and analyzing those with these ELISA kits. And for the higher concentration samples above 10 micrograms per 100 gram, we validated those concentrations with HPLC in a collaboration with NOAA. So in our first paper about this in harmful algae, our initial impressions were that saxitoxin was commonly detected across all categories that we had tested, including about 1/3 of die-off MERS, live MERS from these breeding colonies, and kittiwakes, as well as forage fish. So it was not clear that saxitoxin was implicated in this die-off, but it was notable that the concentration of saxitoxin was higher in die-off MERS than it was in live MERS. And we later learned that there had been a big bloom of Alexandrium catenella in 2016, right in Cook Inlet, near these breeding bird colonies. So our kind of baseline control of presumably healthy live birds might have had a higher concentration and detection probability of saxitoxin than in a normal situation. So we left that study with more questions than answers, and that really spurred more studies that have been ongoing for the past decade, including dosing studies, food web sampling from both die-off events and harmful algal bloom, as well as live bird sampling. And since then, the stakes have gotten higher. The Woods Hole Oceanographic Institute scientists have found massive concentrations of cis in the Chukchi Sea, really at unprecedented global levels. And Don Anderson's paper in 2022 showed extremely high concentrations in 2018 and 2019.
1 second, Sarah. We have someone that unmuted. 1 sec. Let me catch him.
Thanks, Dave. Okay. And then following that, in 2022, E.V. Fashan noted the highest concentration of Alexandrium catenella cells than have ever been documented in polar waters in that same region with the high concentration of cis. And in Kathy LaFay's paper in 2025, she also found very high cell concentrations down south in the Bering Sea near the Pribilof Islands. And the cycle of marine heat waves and seabird die-offs also continued. So in this figure on the left, the top panel shows the heat wave sea surface temperature anomalies throughout time in the northern Bering and the Chukchi Seas on the top panel, and the Gulf of Alaska on the bottom panel. And you can see within that red box the anomalously warm sea surface temperatures have really increased in the past decade more than before. And in conjunction with that, the seabird die-offs have also increased in frequency, duration, and spatial extent in Alaska across the last decade. So this brings us to our main questions. Are saxitoxins responsible or involved in these seabird die-offs? And in order to answer that, we're going to have to ask a lot more questions. Is saxitoxin commonly found in Alaska seabirds and prey? Before this work, we really didn't have a good handle on that. How do concentrations of saxitoxin in birds compare to those in the environment? What saxitoxin concentrations are harmful to seabirds? What tissues harbor the highest level of saxitoxins, and how long are they detected in birds? Are saxitoxin levels in birds that died from saxitoxin higher than others? And finally, could people get paralytic shellfish poisoning from eating seabirds? So to answer those questions, I'm going to use a bunch of different data sources, including data from our Alaska Science Center research, from food web sampling across a range of saxitoxin concentrations and during seabird die-offs, as well as live bird sampling, both from Alaska Science Center studies and contributions from lots of different collaborators. Also, a collaboration with the National Wildlife Health Center, where birds that are submitted to their lab for testing and pathology get sent to our lab for testing for toxins, as well as dosing studies of both mallards and common murres in captivity, dosing with saxitoxin, as well as publicly available or published data that kind of fill in some of the gaps. And at the bottom, you can see the link to our USGS Alaska Science Center data release for our toxin results. And this is a dynamic release. So as we have new analyses ready and have them QA/QC'd, they quickly become part of this new data release. So check it out. We're going to have a new release soon with a lot of new data added. And just to give you an idea of kind of the size and scope of these samples, they span a lot of areas of coastal Alaska. We sampled 58 different events from 2011 to 2025 that I'll talk about later on here. And they spanned a range of saxitoxin levels, die-offs of seabirds, marine mammals, and fish, and also live samples from seabirds. So diving into the first question, is saxitoxin commonly found in Alaska seabirds? Yes, it is. We tested 43 different species of seabirds and found saxitoxin was detected in 42% of those bird species. And I've got a red dashed line for that human regulatory limit for saxitoxin, 80 micrograms per 100 gram. And you can see that only one species, black-legged kittiwakes, approach that human regulatory limit. Is saxitoxin commonly found in Alaskan seabird prey? Yes, also yes to that. We tested for saxitoxin in 27 different species of forage fish and found 78% of those had positive detections. And Pacific sand lance, which are really important fish for lots of different marine food web members, had the highest concentrations, including concentrations above that human regulatory limit of 80 micrograms per 100 gram. All right, moving on to number 2, how do concentrations of saxitoxin in birds compared to the levels in the environment? And in this graph, I'm showing saxitoxin concentrations +1 log transformed on the y-axis, and that's because the concentrations are so variable between mussel concentrations at the highest to really low concentrations in the birds, so this is the best way to show the data. And then I have the different taxa: shellfish, primarily mussels, fish in the middle, and birds on the right. And I've organized these into 2 groupings. These are events that had both mussel samples and bird samples from the same time frame, location, and date. And those that had mussel concentrations above 80 micrograms per 100 gram were organized into the high event magnitude category, and the ones with shellfish levels below 80 micrograms per 100 gram were in the low category. And you can see that mussel concentrations are the highest, then fish concentrations are the next highest, and seabirds are the lowest concentration of saxitoxin. And across these events, we see that the concentrations of in the birds, fish, and the fish are higher during the high magnitude events and lower in the low magnitude events. So yes, the concentrations of saxitoxin in birds are comparable to the environment and predictable in that way. Moving on to the dosing studies, we can see from those dosing studies that what saxitoxin concentrations are harmful to seabirds. So the mallard study and the murre study found a lethal dose of saxitoxin, acutely lethal dose of 167 micrograms per kilogram in the mallards and 366 micrograms per kilogram in the murres. And in the murre study, they also calculated an effective dose, which basically tells at what concentration of saxitoxin are the bird behaviors changed enough that it would likely lead to death in the wild. And that concentration was different depending on the type of saxitoxin ingested, between 89 micrograms per kilogram to 366 micrograms per kilogram. And so the concentration of saxitoxin that is harmful to birds depends on the size of the bird and the size of the prey or meal. So for example, in the murre study, for an approximately 1 kilogram murre eating a 5-gram meal, which is kind of an average prey size for a murre in Alaska, it would need a dose of 1,780 micrograms per gram in that 5-gram fish to change its behavior in a way that would likely lead to death. So what tissues have the highest levels of saxitoxin and how long are they detected? Again, in the dosing studies, we see that the highest concentrations of saxitoxin occur in the GI tract and the feces, but the tissue concentrations were not equatable to the dose of saxitoxin that it was given. And we also found that saxitoxin was not detected for very long after dosing. The fecal samples of birds that survived the dosing experiment didn't have saxitoxin detected after 24 to 48 hours. So that was quickly flushed through the system. And for birds that survived dosing and were euthanized a week after the experiment ended, there were very low concentrations of saxitoxin detected in tissues. And an ecologically relevant finding from the murre study in the chronic dosing trial, where they were fed subacutely lethal concentrations of saxitoxin over time, the birds that survived dosing ate less and lost weight compared to the control birds. So moving on to the fifth question, are saxitoxin levels in birds that died from saxitoxin higher than others? And in this graph, I'm showing again the log transformed concentration of saxitoxin, because there's a really big difference between the highest concentrations in the feces and the lowest concentration in the muscle tissue. And I have different categories of birds here. Dosed birds in red, dose that died from dosing, dosed birds that survived dosing in green, dead birds from the wild in yellow, and live birds in the wild in gray. So you can see that the birds that died from saxitoxin in the dosing experiments did have higher concentrations of saxitoxin present in the feces and the GI tract than other bird groups. But interestingly, the liver concentration of wild birds that had died was higher than other groupings. So just like the dosing studies found, the tissue concentration data is not the most straightforward way to determine whether birds have died from saxitoxin or not, unfortunately. So final question, can people get PSP from eating seabirds? So I want to start by reminding you of the FDA human regulatory limit for shellfish of 80 micrograms per 100 gram. And USGS is not a regulatory agency, and harvesting in Alaska is at your own risk. So I'm showing the maximum saxitoxin concentrations in micrograms per 100 gram across a variety of different bird tissues and different categories of wild birds found alive, wild birds found dead, dosed birds that survived, and dosed birds that died. And for wild birds found alive, we did find about 2 birds had fecal samples that were just over that human regulatory limit of 80 micrograms per 100 gram. But the other tissues, muscle, liver, GI contents, GI tract, and kidney were all very much below that human regulatory limit. Wild birds found dead were all below the regulatory limit as well. And dosed birds that survived did have feces concentrations above the regulatory limit, but no other tissues were above. And the dosed birds that died acutely from saxitoxin poisoning did have concentrations in higher concentrations above the feces, GI tract, and GI contents. And I also want to draw your attention to the percent of quantifiable individual samples from these different groups. So the dosed birds that died acutely had the highest concentration of quantifiable samples, 63%, not surprisingly. Dosed birds that survived only had 37% of tissues that were quantifiable, and wild birds that were found alive had higher concentrations, about a quarter of those were detectable, quantifiable concentrations, compared to wild birds found dead with only 6% of their tissues being quantifiable. And then it's important to compare this to the other parts of the food web that we sampled. So looking at shellfish samples, primarily blue mussels, we see really maximum concentrations of very high, over 11,000 micrograms per 100 gram in shellfish in one of these events. And in forage fish, from one event, these are sand lance samples, the highest concentrations were about 9 times higher than the human regulatory limit for shellfish consumption, in the 700 range there, both whole organisms and GI tract. So to answer the question of whether people could get PSP from eating seabirds, it's not likely, it appears, from what we've seen so far. But again, USGS is not a regulatory agency, and harvesting is at your own risk, so we can never say what is safe to eat. And then going back to that main question of whether saxitoxin could be responsible for or involved in these seabird die-offs, I want to walk you through a few of these events that we know were related to high saxitoxin levels. So starting in 2011 and 2012, there was a die-off of Kitlitz's murrelet chicks in Kodiak, and they had high levels of saxitoxin that was suspected to have killed the chicks because of saxitoxicosis. And then in 2017, there was a multi-species die-off in the Bering and Chukchi Sea including lots of different species of birds, but northern fulmars in particular had higher concentrations of saxitoxin found, and the National Wildlife Health Center determined that some of those individuals likely died because of saxitoxin. And interestingly, there was also a die-off of walrus in that same time frame that also had high concentrations of saxitoxin in their tissues from Kathy LaFay's work. And then in 2019, there was a die-off of Arctic tern chicks in Juneau. There were also some dead sand lance in that same area, and those birds were determined to have died because of suspect saxitoxicosis. And then in 2020, out in Unalaska, there was a die-off of tufted puffins, and there was also a die-off of sand lance at the same time. Unfortunately, the sand lance weren't able to be collected, but the tufted puffin was determined to have died from saxitoxin poisoning, suspected. And there was also, unfortunately, a die-off of an individual who ate some mussels and snails. In 2024, there was a die-off of northern fur seals in the Pribilofs, associated with high concentrations of saxitoxin in mussels. No birds were found in that event that I'm aware of, but there were lots of fish that died at the same time. And then in 2025, last summer, there was a die-off on St. George Island involving multiple seabird species, including primarily northern thick-billed murres, as well as lots of sand lance, and those had the highest concentrations of saxitoxin that we found in forage fish thus far. And there were also multiple marine mammal species, mostly northern fur seals, that died at the same time. And one of the birds that died had the highest concentration of saxitoxin that we've seen in bird kidneys. So to answer whether saxitoxin is responsible for or involved in seabird die-offs, sometimes, although there have been a lot of die-offs without any evidence of saxitoxin involvement. And starvation is probably the most likely cause of die-offs, except for some of these circumstances that I've highlighted. But in general, seabirds are really good sentinels. They can tell us when there's a disruption in prey supply. They sometimes die because of toxin involvement, and they're a really conspicuous, easy to see indicator of what's going on in the marine environment. So some ecological takeaways. The saxitoxin dose for acute mortality found in these dosing studies wasn't commonly observed in prey. That was that nearly 1,700 microgram per 100 gram concentration. But the size matters, both the size of organisms consuming the toxin, so chicks are often more susceptible to toxin poisoning, and smaller fish can be more toxic. So the saxitoxin concentrates in the GI tract, and smaller fish have a larger GI tract to body ratio, so those can become more toxic. Sand lance had the highest saxitoxin of the forage fish that we tested by far and away. And the dosing studies showed that saxitoxin depurates really quickly from the birds. But the concentration in the birds varies with environmental levels, so a detection in a bird can indicate a recent exposure in the environment. These saxitoxicosis events tend to be localized and small scale compared to the very large scale die-offs that are often because of other proximate causes like starvation. But it's important to think back to the dosing study that found that birds that were dosed subacutely lethal levels of saxitoxin lost weight and weren't consuming as much prey as other control birds. So the effect of those sublethal concentrations of saxitoxin by birds could be underestimating the effect of saxitoxin on birds. And the weight loss and rapid depuration could appear to be starvation. So it's likely that saxitoxin is at least an additional stressor in a lot of die-offs. So looking forward, in 2026, this summer, we're forecasted to have a super El Nino, and that is likely to affect a lot of our oceans. And prey distributions, die-offs, and HABs often occur alongside warmer waters. And Kathy LaFay's recent paper showed a clear link between saxitoxin increasing in the Arctic in marine mammals alongside ocean warming. Die-off causes are often initially unknown, but they're often resulting from multiple compounding stressors. It can take time to rule factors in and out, and we really rely on community observations and sample submissions. So I want to thank all the community members and agency folks who have contributed samples. That's really vital for the work. And if you have questions and feedback about what you want to know about HABs moving forward, I'd love to hear it. And if you see sick or dead animals this summer or anytime coming up, tell your local environmental contacts. And you can also contact the Fish and Wildlife Service Sick and Dead Bird Hotline listed there, or the AHAB Network website has a lot of great information too. And I have a lot of people to thank for contributions, sample submission and collaboration, funding, and in-kind support. And I've also listed that link to the data release that I was talking about earlier if you want to follow up and find some more data online. And that's all. With that, I'll take questions if I have any.
All right. Well, thank you very much, Sarah. This is the time that while everyone's thinking of their questions, this is the time for the Straight Science audience to show some love for the speaker. Everyone's got their emojis. I hope you can see that on your screen. And also, it's nice to drop something in the chat because we know it's never easy to be a public speaker, especially after you've probably had a long day. And here you are giving it to us, all this good information. And so I know I've got a question or two, but we'll open it up to the group and see what we get. We've got a question. I'm having some real interesting stuff with my screen. Do we have a question? I thought I saw a hand raised on there. So one thing I was going to mention on that 2017 event, I'm going to see if I can drop a little something in the chat. And this might provide more because walrus, it's an important thing. We did not have high concentrations. We knew we had walrus die off along a beach in 2017. A walrus is a really important animal for the Bering Strait region, so I want to make sure we get this one at my end right. So in 2017, though, a walrus was harvested at Diomede, and its behavior was unusual. And because of its unusual tired, I got the call, and it was this animal had been really tired, and it had come out of the water with people around it and had put its head down and whatnot, was not responding correctly. Anyway, one thing led to another, and that was when that animal, the people sent samples out of concern, wondering, can you tell us why? What happened here? Very unusual situation. And I believe also at that time there'd been several floaters, but it had been awful rough to go get those. So it had calmed down enough that, walrus were in the area, and this one came up, a big adult bull, not acting correctly. So anyway, it turned out, due to Kathy, the concern was from the village. The Diomede took the samples, sent in the samples. We got those samples to Kathy LaFay's lab, where she found it was 5 times over the seafood safety limit in the feces of that adult walrus, and that started a whole thing. Concurrently, at that time, we had that 37 or so, I can't remember, it's probably in that thing there I just put in, dead walrus that were along the beach from Wales to Shishmaref. So the north shore of the Seward Peninsula, all waters kind of roll north for the most part through the Bering Strait, and you have 2 big eddies that kind of go on either side of the strait, and that if there's stuff floating, it often can be found on that northern shore of the Seward Peninsula. It's a great place to find debris and problems, right? So along that coast, there was, at the same time as that, what you might say a hot walrus, or a walrus that had a high level over the seafood safety amount, we weren't able to sample those, just the logistics. We were able to get on, I think, anyway, read the thing. I don't want to take away your thunder. But anyway, just know that for that was local response.
Okay.
And that particular walrus that was very elevated and started everyone like, huh? We have this problem? And, whatever it was, 5 years later, right, we have the big bloom event of 2022 that hit
Mhm.
this region. Extraordinary for us, extraordinary as they're telling us. Those are not only national record levels for Alexandrium catenella, the cell counts, but they were
Mhm.
like global record
Yeah,
amounts. And
that's
so
wild.
that, and it is, and it was the most persistent, most toxic. So that put the
Mhm.
real, that's why we're so glad to have researchers like yourself studying this and presenting to us tonight, because it is a topic we're learning about throughout the region and we're pretty worried about because it just keeps getting more conducive, and the news about harmful algal bloom seems to be um, food for thought.
Yeah. Yeah. Thanks for your perspective and thanks for all your work in submitting samples because that is
Well,
invaluable.
it's to the people here throughout.
Mhm.
It's the people from the communities. I'm a public servant of the region, but the food security conservation concerns for our uh, regional constituents is high. And we have a
Yeah,
lot of other problems, of course, but harmful algal blooms, anything wrong with the marine ecosystem or any of our ecosystems is a big concern, big concern.
for sure.
So, so I hope that helps with that. Any questions to Sarah? I was interested in the sand lance. Now,
Mhm.
when you showed a map a while back, was that where all your samples were collected, or were those sand lance from the Gulf of Alaska? I missed it.
So I didn't talk too specifically about them. Those sand lance were from St. George last summer.
So
So,
Bering Sea.
yep, yep. Trying to figure out how to go back now. There we go. Um, yeah, they were that's 762 and 778. So all the sand lance,
Goodness.
I actually have a table at the end that I didn't present, but I thought I might get a question about the sand lance concentrations and the rest of the concentrations around the St. George mortality event. So we analyzed samples from live birds and found 50% of the birds had saxitoxin detected, but the concentrations weren't super high, which isn't that unusual for birds. Dead birds, a quarter of them had saxitoxin detected, and I mentioned that that highest concentration was found in the kidney of a parakeet auklet. There were a couple gattids, pollock, and an unidentified one. Um, they both had saxitoxin concentrations detected, and those were approaching the human consumption limit, about 50% lower, or half that of the 80 microgram limit. And then those sand lance, every sand lance we tested had detectable
Wow.
concentrations, and the mean concentration was about 200 micrograms per 100 gram, and the max was of a whole fish was 762. And then
Wow.
the mussels were hotter than that, 60% higher than the human regulatory limit. So that was a hot event, and lots of
Yes.
things died.
Yes. And that makes me think about our ecosystem to the north here, where Pacific sand lance is, it's not just birds, right? It's mammals as
Yeah,
well. So
yep.
that's
Yeah, and I'm not showing marine mammal results, but Kathy LeFay has shared some of those in some meetings already.
yes.
But she's mentioned that the tissue concentrations that she found in the northern fur seals are the most clear cut link to saxitoxicosis that she's seen in the marine mammal world. So this was a hot event, and it's kind of unfortunate and also telling that the bird results, we didn't have that many birds to test, but the birds just don't hold on to toxin quickly for very long. So we didn't see that many concentrations, but in the few 8 samples that we tested, one of the dead birds had a pretty high concentration, but it's just really hard to catch it in birds.
And I'll swing back. I have more questions on that, on how birds may be dealing with that or alerting to that. But Doug Causey has his hand up. Go ahead, Doug. Floor is yours.
Hi there. So Sarah, given what we think is going to happen this summer and possibly next summer, are you aware of any organized sampling or surveys to see what the current saxitoxin levels and others might be, or is this more adventitious that when someone sees dead birds on a beach, then we know that there's an issue?
Yeah, I've been working with collaborators for the uh, kind of the live bird component for quite a while. Middleton Island folks, every time they handle a bird, they've been collecting a fecal sample that I test. And then, of course, when the die off happened on Middleton in, was it 2023, 2021? Time is irrelevant. uh, we were able to test those birds for toxins too, but and then the Maritime Refuge folks, especially in the Pribilofs, St. George and St. Paul, have been collecting fecal samples on the birds that they capture and collect too. So we kind of have we're developing a pretty good baseline over time from some of these locations. And then any of the birds that we collect in my studies in Cook Inlet and colony birds, we've been collecting fecal samples. And if we have a dead bird, we'll test them. So it's kind of regular in a few colonies and then kind of opportunistic, but yeah, you also have to be there in time to catch a bird before they deteriorate. And, these samples, they get old and decomposed, are not as useful. So it's kind of timing and luck, as you know.
Right. Thank you.
Comment on that. I don't see anyone else yet raising their hand, but somebody jump in. So on that, I understand those are when you have field camps and research and it's sort of like it almost sounds like the cart and the horse, is it that there's a harmful algal bloom and you sample the birds, or you got dead birds and then you go sample the war. So for the coastal communities in Western Alaska or here in the Bering Strait region, we don't have any federal marine resource or land resource for the natural resources in the hub of Nome in the Bering Strait region. So it's really on us. I know that since 2022, there's several research efforts that are passing through the strait, and some we know are looking for harmful algal bloom, some we don't know, and they're probably looking for or seeing things in the water column. We just don't know. Is at the bigger agency level, is there thoughts on how to provide service more to remote communities? And I know that's a big ask, but is that something that comes up in conversation at your end? Because people here are utilizing the bird for food, seabirds for food, and there's a human health potential component for that. And it may not be in your wheelhouse to answer that, so don't feel under pressure to answer. But do you hear any more than usual of like, boy, we really hook in with?
Yeah, I haven't heard things outside of my research group, but
Yeah.
I've been super interested in pursuing that aspect. And it's one of those things I feel like we need to write a proposal and get some money, dedicated funding for. But I think it would be really awesome to work with communities that are worried about subsistence seabirds, worried about toxins in them, and try to figure out exactly what tissues are the most important, what species, how different preparation methods might affect the toxicity, and kind of come at it from the community aspect of what's the biggest concern, what organisms are you most interested in learning about? And I could envision a lot of different ways that we could hopefully be helpful. We're never going to be as quick as the DEC for commercial harvest, but because it's a research program, but hopefully over time that would be a great collaboration to try to, if community members wanted to save subsistence harvested foods and have them tested, we could hopefully get an idea of concentrations throughout those organisms, and it wouldn't be real-time information, but it'd be something.
Right. And no, as a model of that for out here, I know it's not federal, but we do have our state Alaska Department of Fish and Game, which is here for like land animals, right? The moose, the caribou, and fur bearers, and then also we have commercial fisheries. So there is some service in the region. People are used to, say you shoot a moose and it's full of warts or bugs or whatever, something's not right. You cut it off and you can come into the office in Nome with the hub, and then there's a that gets sent off to the state vet. We just don't have anything like that in the marine environment currently for the marine resources,
Yeah.
like a seabird. And so I hope it's not yours to fix, but I hope that that's becoming more awareness, that people have expectations maybe of, especially when things go wrong,
Yeah.
so hopefully that'll be something that in a perfect world, we'll have
Yeah.
more access to. But if it takes getting a special grant for doing that, then let's do it, because we need to get answers, So I really
Yeah.
appreciate all that you're doing, the work that you and your group, your USGS group does on birds, and like presenting this information to us. It's fantastic.
Yeah.
More than
Thanks for
we have
having me.
heard. So we really appreciate your time on that. And let me check the chat box. So circling back around, do the birds seem to, since the birds, you can't seem to catch it in a high amount, and maybe it's not there. Are the birds able to, is there any thought, can they detect it, or do they physically, I don't know,
Yeah.
react to it, or have
Yep.
some other sensory?
Yeah, it's a great question. From the dosing studies, it looked like both mallards and murres were regurgitating prey that was too hot, either, well, in the
High
mallard study,
concentration.
yeah, so in the mallard study, they gavaged them with purified saxitoxin, and then in the murre study, they gavaged in one study, and the other study, they injected forage fish with toxin and then fed the fish to the birds. And the birds, if they they ingested it, and then some of them quickly regurgitated, and sometimes it allowed them to live, and then sometimes they weren't able to regurgitate quickly enough and they died. But they either regurgitate and often then don't want to eat anything else, and that's probably why they're losing weight in these situations where they're subacutely lethal levels, which, theoretically, if there was a bunch of prey without saxitoxin available, that wouldn't be that big of a deal. But in reality, if there's a bloom of Alexandrium, you're likely to see concentrations in a lot of the food. So that could easily add up to poisoning that might look like starvation.
Right. Yeah,
Yeah.
birds got a high turnover metabolism. And just for the
Right.
audience's sake, when you say lavage, the poison was put directly into the stomach. Is that right?
Yep. Yep.
Yeah. Some people may not know that word.
Yeah.
All right. And I have to say, you have some irony on your Super El Nino slide, because it's under looking forward. Like I thought, oh no, we're not looking forward to the Super El Nino. Do you have any, what's anticipated, do you know, for our region for the Super El Nino?
I don't know too much about it, except it doesn't look good.
Okay, it's coming our way though. Is that what you're saying?
Yeah. Yeah.
Yeah. All right. All right. Any other bird questions, concerns? You got it. You nailed it, because you've got some bird people on the line here, and they are either stunned and amazed, or
Ready for dinner.
knew it. What's that?
Or they're ready for dinner.
Or they're ready for dinner. And so maybe that's it. Any other questions? All right. Seeing none, we will let everyone get back to dinner. And we would thank you very much, Sarah. And if you could stay on for a minute, that would be great afterwards. Yeah. And next week, we're taking a break. So there will be no Strait Science. However, the next Strait Science will be on 14 of May, I believe. And that will be the North Pacific Research Board's Danielle Dixon will be giving an overview of a 5-year multidisciplinary project that they're doing in the Northern Bering Sea, Bering Strait region called the Northern Bering Sea Integrated Ecosystem Research Project. So we'll hear from this project as it unfolds, but we'll get a big overview from Danielle Dixon and the North Pacific Research Board, which is funding a large portion of it. So it's a big multi-year study. I don't know if you're involved, Sarah. There's so many people. We'll hear. No, all right. We'll hear about it on the 14th. With that, thank you all so much for coming, and we'll see you in May.